Hello everyone, welcome to our paper trailer video about time-dependent image generation of plants from incomplete sequences with CNN transformer. For phenotyping in modern agriculture, we are often interested in the development of crops over time. Therefore, we fly over a field by drone several times in the growing season, and when we look at the same position over time, we get a multi-temporal image patch, as you can see here in the upper line. But in almost all cases, this image sequence is incomplete with missing images in between. This means, although there were overflights, for instance at day 47 or 67, the images are partly not usable because of blurrings, deformations or artifacts. Those distortions are often related to weather influences, for example when clouds move in or wind blows up during a drone overflight, and thus are pretty hard to avoid. As a result, all tasks aiming to analyze the temporal evolution of crops become much more difficult. Our idea is to replace missing images by images generated with a neural network, so that we have, in the end, really artificially generated sensor data. For this, we need a model which can be trained on several incomplete sequences like these here. So if we then only have, for example, 3 input images at the bottom with pink frame, we are able to generate realistic estimates for all the other time points. This means these are then both interpolations if the newly generated images lie in between the existing images, or extrapolations if they are outside this range. The model we propose is called TransGrow and is a conditional generative adversarial network. Without going too much into detail, the important thing is that we combine convolutional with transformer layer and positional encoding. Especially the transformer, with its ability to process sequences of different lengths, is responsible for the fact that we can actually take an incomplete time series in the input and make it more complete in the output. We are also able to generate a distribution for each generated time point, which allows us to visualize the variability of the generation process. Looking at some qualitative results from the lab plant Arabidopsis, where we have generated new images from only 3 input images in the bottom, we can see that the structure of our plant remains consistent over time and also fits well with the reference. Unlike baseline approaches, our generated images are not blurred, no artifacts appear, and it is possible to perform extrapolations. Also, the variability maps are more reliable with more variance on the leaf edges where the plant actually grows and less in the background. For the mixed crop dataset in an even more complex outdoor environment, we have similar findings. TransGrow perform better than the other approaches and can also handle different lighting conditions in most cases. If you are now curious how flexible time-dependent image generation of plants works in detail, please just have a look at our paper or the code and the data, which is all open access. Thanks for watching.